I'm going to talk to a large extent about the nervous system. I'll give you a little bit of stuff about the brain at the end, if I have time. But these organisms in the lumen on the left actually do a lot of things. They make short-chain fatty acids. When they ferment, they form gases. All these gases are neurotransmitters. Carbon monoxide, nitric oxide, and hydrogen sulfide. They're all neurotransmitters in the intestine, and they all have very significant effects themselves. But the effects on the nervous system, not just in controlling the local motility and so on, also affect the immune system. And I'm not going to talk much about basically about that, and rely largely on the nervous system. Now you can recall from the mesenteric nerve. And my colleagues, where I work, this is Wolfgang Kunz's work, actually has recorded from the mesenteric nerve that actually takes all of the messages from the gut up to the spinal cord, basically in the brain. Each one of those, what looks like noise, each one of those is a neuron that is firing. And in fact, we've recently shown, I'm not going to give you the data, that Lactobacillus, and including Lactobacillus reuteri, actually can suppress, to some extent, this firing, which is very much to the point. We have studied pain simply by blowing up balloons actually we've done this in humans, as well as in rats. But so we would prefer rats because they're anesthetized and they don't complain. And so this is the story for the rats. We blow up the balloon, and then we look basically at the left-hand end of things here. The bradycardia response is an indication of pain in this model. And here you can see that if we feed the lactobacillus for 9 days, then in fact this pain is completely inhibited, which is quite remarkable. A single organism fed 10 to the 9th per day for 9 days. The earliest that we see this is already at 3 or 4 days, but we feed for 9 days. So it can completely inhibit this pain response. And when it comes down to it, this pain is what we're interested in. These animals are anesthetized, so we're not looking at the brain and the brain's perception of the pain. We're looking at the local response. Now, another study from some colleagues of mine at Master, the Collins group, this is Eleanor Verdu's work, has shown that if you treat animals with antibiotic, it's a broad-spectrum antibiotic wiping out most of the flora in the intestine, then actually you can demonstrate both an increase in inflammation in the gut, and also you can demonstrate that there is an increased pain response. So the mere fact that you don't have all the organisms present, at least in animals, and this is yet to be shown in man, but it raises some very serious questions about antibiotic treatment in general that I'll leave you with. The nervous system in the gut is very complicated, and the enteric nervous system, which is all of the nerves put together, go up 2 different pathways. On the right-hand side, you can see that it goes up to the spinal cord and the brain, and on the left-hand side, through to the vagus. And so the question in part is, are both of these systems affected, and under what circumstances are they affected? Because that would give us some other approaches to trying to treat visceral pain. We, a few years ago, carried out this study, which is again the animal model in which we blow up the balloon and we record the pain. And in fact, you can see, just for the record, this is the background, and you can see that in fact there are 3 different things in this particular model system that actually prevented the pain. The first was the live organism. The second was the dead organism. I repeat dead because it was heat killed or it was gamma irradiated, and gamma irradiation doesn't do much to the surface components of the bacterium. And the third was the conditioned medium in which these would grow. So something is coming out of the bacterium, as you heard the talk by Maria Racino, something is coming out of the bacterium or being shed by the bacterium, which has some very distinct properties, not immune properties in this case, but activities on the nervous system. Many immune properties, however, are also exhibited. by these organisms. And this is just a cartoon showing you on your right the lymphoid follicle pairs patch, or part of the pairs patch. And the bacteria can affect the epithelium, they can affect immediately the immune system inside the pairs patch and the dendritic cells, which in turn are communicating with the regulatory population, which they upregulate. And you can show, or we have shown, that these particular lymphocytes, once in fact exposed in vivo to these bacteria, these Reuteri, or sometimes we use a rhamnosus preparation, all then start to synthesize the most regulatory component, cytokine, IL-10. Now, IL-10 is, I'm not going to talk about the immune properties, but because they have huge number of immune properties. But IL-10 is in fact, look at the bottom here, IL-10 is an anti-nociceptor, or prevents pain perception as much as it does immune regulation. And we think that this is just a part of the whole business of regulating pain. It's not just direct action on the nervous system. My colleagues can record from the myotonic plexus, and myotonic plexus shows the layers. The lumen is to the bottom of the slide, and you can see the lumen with the villi, and then you can see various sheets that actually unravel, and we can dissect out the myotonic plexus. And then my colleague Wolfgang Kunz, who is the main neuroscientist working with us, can look at this, which is a fanciful cartoon of a patch clamp, in which a very, very small, nano-sized surface area on the surface of the cell can then be patched, sealed, and you can record the electrical activity. And the bottom line in this is, and that's a fanciful patch clamp that shows you in red, one of the nerve cells in the myotonic plexus in green, the green blobs, in fact, in vitro. This is the sort of recording that he gets when he stimulates. And again, the bottom line in this is not to have to explain some of the fanciful, more sophisticated neuroscience in this, but if you read at the bottom, and it's the slide that Professor Stanier actually showed, that in fact, if you feed one of these organisms that seems to work in preventing pain, it actually has a specific action on a specific ion channel. This is so-called IKCA channel, or 3.1, a very, very specific channel. And only those that actually have activated this channel so far in our hands actually have this pain. So we think that it is heavily involved, quite specifically, in this type of model system. We have another system in which we look at motility. And I think it speaks for itself. You have pressure on the left in a bottle. You can raise the pressure, and I'm going to show you, if it works well, a video of this, because we can then feed bacteria down the middle of the lumen of the piece of intestine that we're looking at. And in this particular system, we're now not talking about gut that's been taken out of an animal that's been fed. We're talking actually about a piece of gut from a perfectly normal animal. So these experiments that I'm going to show you clearly indicate that these bacteria can have an immediate action, immediate within minutes, on the peristalsis that is occurring, which is in itself remarkable. They don't have to go through all the other components that we normally would think about. This just shows you that, in fact, on the left, you can see some of these motor complexes that we record, and it can show you that after feeding, in fact, not feeding, excuse me, after putting these bacteria into the lumen of the gut, the actual complexes are decreased in size, you can see that, and in fact they are reduced in frequency. And this is actually, amazingly, it has a dose-response effect. You can see the dose response on the right. It's like a pharmacological effect. And it is a pharmacological effect for all intents and purposes. What we're obviously interested in is what is actually causing that pharmacological effect. We have some clues, but we don't have the answer. So this is actually from you, Flavia. This is your slide, so you better look at it and just tell me if it's okay. But I mean, this is Flavia India's slide showing that the Rotary can improve gastric empty in healthy preterm infants. And we're able actually to look at pain in the stomach. I'll just show you, just because I'm changing gears to talk a bit about the stomach. And what you see is actually a slide showing the bradycardia that occurs in the stomach when you dilate the stomach with an increased pressure inside the stomach. And exactly the same thing happens as happened with the colon. And in fact, this mirror image is a mirror image showing you that the bug that I used at the beginning actually has the same effect on the stomach as it does on the colon. However, when we tested a whole bunch of different organisms, more than shown on this slide, you can see that, in fact, many of them, the salivarius doesn't work on either. The Bifidobacterium infantis, which is the Align product from P&G for IBS, that only works on the colorectal and doesn't work on the stomach at all. And the plantar is another mutant that we'll talk about maybe some other time, also has differential effects. Main point, probiotics are not probiotics are not probiotics, every effect is strain dependent. It's got nothing to do with the fact that the bacteria are or not present in the stomach or bound to the stomach lining. They clearly are, as shown here on two of these organisms, one which works on the right and one which doesn't work on the left. So I wanted just to finish up with a couple of a few slides, just demonstrating that in fact there are many other things that these bacteria can do, which are highly relevant to functional disorders. And the first of these is that in fact we have shown, and others have shown as well, that some of these bacteria in fact can completely attenuate the normal HPA axis response. This is corticosterone. The animals are being acutely stressed. In this case, they've been fed these bacteria, and you can see on this, basically on the slide, that the organism, when fed, in this case for 28 days, but we've done it for 14 days as well, inhibits the level of corticosterone response to acute stress. So stress can also, to some extent, be inhibited. The question is, how does that occur when you feed a live organism into the lumen of the bowel? How does this occur, and how does this effect translate to the brain?